saying welcome to the second presentation of this Unisense workshop with focus on biogeochemistry. Since I'm the biogeochemist here, it's me who is doing the presentations. Yeah, so this is the second presentation of today. Talk about the lab-based studies. And so in this presentation, I will talk a lot about microprofiles. Talk about how what experimental design should be, how you should think about to get good microprofiles. Some examples from hydrogen dynamics in microbial mats, and something about also the electric potential generated by cable bacteria. Then I will talk about the photosynthesis measurements using the light dark shift technique, which is a really strong technique for this. Then a bit about microprofile theory. I will keep it short today. Tomorrow there'll be a longer and more detailed theory part in the last presentation of the day about the, how to use the software to calculate activities from microprofiles. I'll present the microrespiration system and finish off talking about lab and field measurements and how they can interact. And tomorrow I will discuss present about the field measurements in situ measurements. So microprofiles. Well, this is our lab microprofiling setup. What you will need is first of all a microsensor. You'll need an amplifier. We have different amplifiers. We have the multi-channel ones here, which I already mentioned in the last presentation. Those that can both operate the optical oxygen sensor and the electrochemical sensors. We have them in different versions, and then we have the dedicated ones, so they are dedicated to one type of sensor only. Okay, then you will need a micromanipulator. That's the one here, so you can use it as a manual one or motorized one. And the differences are that the motorized one that's shown here, there's this linear motorized stage controlled by the motor controller and the software that can move the whole micromanipulator up and down. In this case, 10 centimeters. We also have a 30 centimeter one that can do this. Do it in steps down to half a micrometer. Not that anyone needs that, but it can do it. If you want to use the manual only, then you don't need this linear stage here. You just need the manual micromanipulator. Then you will use the micrometer screw here to move the sensor up and down in steps down to 10 micrometer at a time. Then you'll need software as shown here. This will operate the motor controller and drive the sensors up and down. It will do automatic profiling, so you just specify what steps size you want to work with and so on, and then it will do it all by itself and collect the data, and it will allow you also to do the calculations, which I'll show tomorrow. So to get nice profiles, that's always what we want. Profiles are nice because these dimensions, that diffusion makes sure that they're always smooth gradients if you measure them right. So you need well-defined conditions. You need a setup that's easy to work with so that it is safe for your sensors, so you don't break sensors by accident. And then steady state is an important part of this, and I'll get into that a few times. And what you get is a microprofile. Typically, you have the vertical x-axis, a horizontal y-axis with a concentration, and you measure down through the sediment like this. You will have a well-mixed water column. You will have a diffusive boundary layer. That's the layer of water sticking by friction to the top of the sediment, where only diffusive transport takes place. And then there will be the sediment down here. So the desired features for such a setup, well, steered and well-mixed water phase. So the reason for this is that if you have a poorly steered water column like this, it's not steered at all, for example, but there'll still be a little bit of mixing. For example, cooling of the water at the surface due to evaporation will cause dense water that will sink and make sort of a sluggish circulation. That means that the concentration of oxygen, for example, in this case, on the sediment surface will vary over time, and therefore you will not get a stable concentration gradient. If, on the other hand, you keep a well-mixed surface, layer or water phase here, you will have a constant concentration of oxygen above the diffusive boundary layer, and you will get a constant profile over time, so a steady state situation where the profile does not change over time. This will create well-defined conditions, and you get steady state, and then data can be interpreted. That means that you can do a lot of calculations on them that require actually that there is steady state. An example of this could be here. I have sediment in small sediment cores here in water that has been sampled on the sampling site, the bottom water, bubbling with air to keep oxygen concentration close to atmospheric saturation all the time. There's cooling coming through these tubes here down to a metal coil, keeping temperature down at 4 degrees, I think it was. And then there is a Pasteur pipette, a plastic pipette here, where air is blown through across the water surface. So that creates a sort of a horizontal flow of water across the sediment surface, and that creates a nice boundary layer, and it's easy to interpret the profiles. So that could be a setup where you can keep constant concentration, well mixed water phase, and get nice data. Yeah. So then when you do profiles, mixed water phase, then you also know where's the sediment surface relative to my microsensor. There are a couple of ways of doing this. One is using a dissection microscope as here. We actually sell this size setup. It's a very nice microscope on a boom stand. It has a 9 centimeter focal distance, so from the front lens, so there's plenty of place or space in front of the microscope where you can have your sediment cores or samples or whatever, and this the microsensor. There's a camera also, which is integrated with our software, so that you can actually take pictures in the software, and they are stored together with the profiles, and then you have them afterwards, and you know which picture came from which profile. Yeah, that another way of knowing where you are is that where the sensor are is that you can use align 2 sensors together. This is an example of an oxygen and sulfide sensor that has been aligned, so you can get true parallel profiles. We have this type of sensor holder for the micromanipulators where you can mount 2 sensors. The second sensor has a set of controls here, so you can tilt and twist and move the second sensor relative to the first, so you can get the tips of the 2 sensors very close together, so you can get real parallel profiles. This has been done here for an oxygen and a sulfide profile. The oxygen profile, once you know these, it's relatively easy to guess or estimate where is the sediment surface. Is it here, or maybe it was up here? I don't know, but it's just 100 or 200 micrometer wrong. However, if you look at the sulfide profile, it's almost impossible to know where is the sediment surface relative to the profile, because you just meet sulfide at a certain depth and you don't know the distance to the surface. But since the 2 sensors have been aligned before the profile was made, then you know that if the surface of the oxygen profile is here, then it's also here on the sulfide profile. And also use that when they are well aligned to, for example, look at these, if there is an overlap, or if the sulfide zone actually comes up and reaches the oxic zone, or if there is a distance between the 2, which could be Campylobacteria, as I will show. So then you also want proper control of water conditions. It could be, as I showed, in a bucket with sediment cores, you want to have control of temperature, oxygen, and nutrients, for example. Here's an example from Iceland, where one of my colleagues was working in a hot spring where at 60 degrees hot water came out of the slope here of the hill, and created these cyanobacterial mats, very thick and homogeneous ones. They took them into the lab in this flow system here, where they were circulating in situ water at 60 degrees and have the microsensor above and light on it, so they could measure oxygen profiles in light and also in darkness here. So they are measuring on the same conditions as were present out there. I'll show a couple of examples of microprofiles and what you can really get from the microsensors, because they are fast and give a high resolution. This is an example from a coastal cyanobacterial mat in Denmark, where they were measuring with a lot of microsensors oxygen, sulfide, pH, hydrogen, and H2X, the one that's insensitive to hydrogen sulfide. They're also measuring light. We don't sell that. That's a speciality of Michael Kühl and his group. What they saw was the green one is light. So light was penetrating about 1 centimeter into this mat. That gave a high photosynthesis here, so a large peak of oxygen. And the sulfide, the red line here, comes up and meets the bottom of the sulfide zone. This creates a large peak in pH. So they were interested in seeing, one more thing I should mention is that hydrogen was 0 here. That is the black dots along this axis. They were interested in seeing what happens when light is turned off. And they then measured as fast as they could, I believe, all these profiles 5, 15, 30, 45, 60, and 90 minutes after light was turned off. And out here for reference, you can see the two graphs I showed before. So you see this big peak in oxygen that had disappeared totally after 5 minutes. There's no oxygen left down here, and it's back and almost doesn't change over this 1.5 hours. So the oxygen is really fast in adapting in this case. The sulfide, the red one here, was forced down by the presence of oxygen in this case, and then it slowly had the chance to get back, and after 1.5 hours, it's almost reaching the bottom of this oxygen zone. There was no hydrogen present in light, but when light was turned off, then a peak of hydrogen developed here, 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 and so on. And you can see after about 45 minutes, it's reached its maximum, and then it goes a bit down again. This shows the, very well sort of the, first of all, that these sensors are very fast, and that means that you can actually measure these profiles with so short interval. And they give the vertical or spatial resolution that is needed to see these peaks and these very steep gradients, oxygen only penetrating 200 micrometers into the sediment here. So that's a big advantage of the oxygen sensors. They also used them in the same study actually, but in France in a hypersaline microbial mat. They were measuring oxygen in light, and then and also hydrogen, and then they turned light off, and then it's 5, 15, 35, 60, and 120 minutes after they turned off light. Oxygen disappeared immediately from the peak, remained deeper down in the sediment even after 35 minutes. So 35 minutes, and that was a big difference to the Danish side. But hydrogen appeared very fast. Not after 5 minutes, but after 15 minutes, you start seeing hydrogen, and then 35 minutes and so on, you see it's climbing up, and as a very prominent peak here after 120 minutes. They wanted to investigate this accumulation in more detail, so you can also use microsensors for that, of course. So they placed the hydrogen and oxygen sensor at 0.8 millimeters depth and measured over time, about 65, 70 hours. These dark parts here are darkness. The white ones are in light, and when there's light, there's a lot of oxygen, and that disappears immediately when it's dark. But then there's a big increase in hydrogen, which then sort of settles down and becomes stable at a 5 micromolar. Then when light is turned on, there's again a lot of oxygen, and that disappears in darkness. Hydrogen appears, and so on. So this is also what sensors can be used for, and this is relating to the question from the last presentation that I was actually surprised to see there's so much hydrogen appearing here, and that is of course a problem to the standard H2S sensor or the SOLF sensor, which is the one that's sensitive to hydrogen. Okay, just one more example of microprofiles. This is from the electric potential microsensor used with other sensors, of course. This sensor was published in 2014, and we started producing it about a year ago, and it's exactly like the ones they used in this. They had a homogenized sediment core incubating from day 0. After 4 days of incubation, they measured a set of profiles. Oxygen going to 0 very fast. Sulfide is high, but stopping at the bottom of the oxygen zone, and the electric potential is 0. And there's a small peak here, negative peak in pH. 19 days, then the situation is totally different. Now the sulfide here, the black, and the red oxygen has been totally separated, and there's a peak in pH. And the reason for this separation is that they have that the cable bacteria have established. And these cable bacteria, they form long filaments all the way through this layer, and they are able to transport electrons in sort of cables they have inside their filaments. What they do is they take the electrons from sulfide down here, so oxidized sulfide to either sulfur or sulfate, and transport the electrons up to where oxygen is present, and then donate the electrons to oxygen and reduce that to hydroxyl ions, giving this positive peak in pH. And doing that, they create an electrical potential gradient here. It's only 1.8 millivolts or so when it's at maximum, but it's easily to measure. You can see this is 3 profiles, I think, with standard deviations shown here. So it's relatively reproducible. So that's possible to do with the electric potential microsensor. So as I promised in the last presentation, I will talk about the photosynthesis measurement using microsensors and the light dark shift technique. So the assumptions for doing this is that you place your sediment in light or your microbial mat or biofilm, whatever it is, and wait until you have steady state so that the concentration profile will not change over time anymore. When that happens, when that is the case, then the production of oxygen by photosynthesis will, in any point down through the profile, will equal the removal of oxygen by respiration and diffusion away from the site. When you then suddenly turn off the light or remove light, then all the oxygen removing processes, that is respiration and diffusion away from the site, they are not changed because they are depending on the concentration of oxygen, which has not changed. And therefore, the change in oxygen concentration that happens after turning the light off will equal the production of oxygen that was there in light before the light was turned off. And I will show you how this works. In order to do this, you need a biofilm. You need a very fast responding microsensor, the one that has a response time of less than 0.3 seconds. In this case, there's a bit of air being blown across the water surface to create some circulation. And then there's the light sensor, which must be connected to the amplifier, because the light sensor will inform the software of exact time when light was turned off. If you look at a biofilm, microbial mat, or whatever, like this, this is over a long period. This is a 5-minute interval here. So this is about a quarter of an hour here that's been in darkness. Then light is turned on and it will accumulate light at the depth where the sensor is sitting, and then it will become stable. When you turn light off, it will decrease very rapidly and then level off again. But you don't need so long time. Actually, we need a very short time for this type of measurement. The software does is that when you use this, it will collect data continuously at a very high rate. When it then detects that light is being turned off, it will save the data from 3 seconds before the light was turned off. This is where light was turned off, time 0, and until 3 seconds after light has been turned off. And then it will find the initial slope of the decrease of oxygen concentration here. The initial decrease here will equal the production of oxygen that was there before light was turned off. So this is actually the gross rate of oxygen production or photosynthesis in that exact spot where the sensor is sitting. So what you can get from it is data like this. The green bars here indicate the gross oxygen photosynthesis oxygen production that has been measured by the light dark shift. Those are the green bars. Now, you can also calculate the oxygen flux away from the photic zone. So these are the 2 red arrows here, up and down, J up, J down. And if you add the 2, then you get the net oxygen production. And that is the net photosynthesis. So you have the gross photosynthesis here and the net photosynthesis here. And if you subtract the 2, then you'll get the respiration rate in the photic zone. So you'll get a respiration measurement in light while you still have the high oxygen concentration. So this is actually the only way you can get this respiration in the photic zone. If you would normally measure respiration, then you will turn off the light and wait and measure the oxygen consumption by the biofilm. But at that time, the oxygen concentrations in the biofilm would have changed a lot, would probably just have been a short profile like this. So this is a really handy technique to use for this, and gives information that you can't really get in any other way. One more example of this light dark shift technique. These guys were interested in knowing the effect of immersion, so inundation or immersion of microbial mat that was growing in an intertidal area. They put it into this aquarium where they could either have water above their biofilm, or they could have the biofilm exposed to air, so water above. And the data are here. The upper one shows the situation where there's water on top of the biofilm. The bars here shows the gross photosynthesis measured by the light dark shift, and this is the oxygen profile measured at 100, 400, and 800 micromole photons per meter square per day. And as you can see, at higher light intensities, the max rate of photosynthesis increases quite a lot when it's underwater. And also the thickness of the photic zone increases quite a lot. When there's no water on top of the sediment here, you can see that oxygen production of photosynthesis it increases a little bit, but not very much with increasing light intensities. And it also does not penetrate as deep into the sediment. So that is an example of what you can measure with the light dark shift technique. So about profiling. So I already mentioned we have different ways of doing profiling. This is the laboratory setup. We have the field microprofiling system, which is all shown here, and we have the in situ mini profiler, which can be taken to the seafloor down to all the way to 6 kilometers water depth. All profiles measured by these can be analyzed by the software. Yes, so on our website we have an application note, the profiling application note, which explains how to use this this facility of the software. You go to knowledge, application notes, and sediment profiling analysis, and then you find this PDF file, and I will go through that tomorrow at 4:45 Danish time. Now I'll just mention briefly a little bit about the microprofiles theory. This is an example where you have biofilm or sediment where the well-steered water face above. You see the vertical profile of oxygen here, so oxygen is the same all over. Diffusive boundary layer with a linear gradient, and then the sediment with the oxygen in it. So what you can get a qualitative information from such a profile, that is up here, we have the straight line. That is a straight line means there's no production or consumption, only diffusion. So that's the diffusive boundary layer. In this part of the sediment, we have the concave profile, so curving this way, and that indicates production of oxygen. It's shown here as a negative consumption rate, this bar graph here. On here, number 3, you have the convex shape of the curve going this way. That indicates consumption of oxygen. That's shown here as a consumption rate. But that you can get from sort of qualitative information, but you can also do calculations and get a lot of quantitative information. Here's an example, sort of a theoretical example of a profile. Well mixed water phase, diffusive boundary layer with a linear gradient, and then there's sediment biofilm here. This has a layer of inactive sand on it, so there's a part of the sediment here that is not active, as indicated by this linear profile here, indicating diffusion only, no production, no consumption. Down here we have the curvature that indicates consumption of oxygen. So the quantitative information you can get from such a profile. First of all, you can get the concentrations. If you are interested in organisms living here in the sediment, you can see what concentrations are they actually exposed to. If you have several profiles of different solutes, you can investigate overlapping zones and so on and see what the concentrations are. Can also do is you can calculate fluxes. You can calculate the flux of oxygen into the sediment here in the diffusive boundary layer by multiplying the slope of this of the profile here, the linear part of the profile, with the diffusion coefficient of oxygen in pure water. Then you'll get the flux of oxygen into the sediment in this exact point where the profile was measured. Also analyze the curvature of the profile down here. And then you can get the consumption rate of oxygen. To get that, sorry, I should start up here. You can also calculate the flux in this inactive layer of the sediment. To do that, you still need the slope of the linear gradient here, multiplied with the diffusion coefficient, sorry, the diffusivity and the porosity. And the porosity takes into account that not all of the volume here is water. Some of it is particles, and you cannot, there will be no diffusion through the particles. It has to get around the particles. The diffusivity here is in its simplest form calculated as diffusion coefficient times porosity. But you can do it in different ways, and I'll get into this tomorrow in this demonstration. There are different ways you can do it, and you can choose different methods in the software. Once you get into this part, you can analyze the curvature. This is also what the software does. You have to get the second derivative of the profile here. So you need some kind of mathematical formulation of the profile, so you can differentiate it twice to get this value. Then again, multiply by diffusivity and porosity. Then you get a consumption rate volume of sediment. So typically nanomoles per cubic centimeters per second. And this can be done in the software. So enough about profiles for now. I will get over to the microrespiration system. So that is a system designed to quantify metabolic activity of small organisms. And these organisms may be microorganisms, or it can be nematodes or copepods or whatever organisms that can fit into one of these microrespiration chambers. The system consists of the chambers, of course, shown here, a small glass chamber with a glass seal and a capillary opening where the sensor can be inserted through. This rack where the sensors are placed has a steering, magnetic steering built in all these spots where you can place the chambers. There you can have a magnetic steer bar inside the chamber. This is glass coated, so it doesn't give off or take up any gases. There's a glass ring around this one, and there's a stainless steel mesh on top of it. This is if you want to have an organism in here that you don't want to be destroyed by the rotating magnet down. And if you work with microorganisms, you don't need this, then you'll just have the magnet. Steering controller that keeps this steered, and then there's a software that keeps track of data and calculates rates. So I will briefly explain. This is a drawing of the chamber. You have the steering down here, you have the glass seal, and you have this glass capillary where the sensor can be inserted. The capillary is only 0.7 millimeters in diameter and 15 millimeters long. And since it's a capillary, water will always be stagnant in here, so no convective transport. So if you have, for example, a low oxygen concentration in here and you have high oxygen out here, the diffusion of oxygen through this long, thin capillary will be so low that it cannot be detected in here. So you can don't even need to check for it. You can under control, of course, but you don't even see it after days of incubation. There's no oxygen coming in through this capillary. But you can insert the sensor through this, and you can do this safely because the sensor is placed inside this blue guide shaft here. And when it's placed in the rack with this plastic tip in the top of the lid, automatically finds its way into here. You can have up to 8 of the chambers of the small size here, and you can move the sensor around and measure in each of them at different times, and the software will help you and keep track of where the data belongs to. So data measured in chamber 1 will be saved as chamber 1 data and chamber 2 and so on. And you can use different types of or different sizes of these chambers. We have them, the small ones, we have from 400 microliters up to 4 milliliters. So these are the ones. And we have medium-sized ones, 20, 40, and 50 milliliters. It's only the 40 milliliters fits in the rack. Then you can have 2 of them. You can have 8 of all these small ones. And then the medium-sized ones, 20 and 50, and the big ones here, 240, 400 milliliter. You will need to make a construction of your own so you can place the sensor on top of it. You can use any of our sensors for this, both the optical and electrochemical oxygen sensors and all the others. It's a complete system with glass chambers, steering, rack, sensors, and so on. The software will show real-time data, and it will collect data for each chamber individually, and you will get graphs for each chamber. I'll just show how the software looks. It's the ones that's called rate. Here's only one chamber, so yeah, anyway, here data have been collected, the green dots here, then there's a break here, and then another collection of data, and a break, another collection, and so on. The software will continuously calculate the slope of the change in concentration here. It'll also calculate R² and standard deviation, so you will all the time be able to see the statistics of your measurements. You can stop when you think it's good enough and move to the next chamber. Then you can analyze the data. So this will automatically fit a line through all data points. What you see in this case is not really smart. But what you can also do is you can use the rates within part here. So for each chamber, it will analyze the slope of the parts where data have been measured. You can also use the rates between, and then it will calculate the rate of change between two measurements. I'll show in the next example where this is smart, but if you have many chambers, you'll just have short periods of measurements and long periods of breaks between the measurements, and then that is really smart. As shown here, where the system was used to measure respiration rate in this small jellyfish. It's a 3 to 5 millimeter diameter jellyfish. It was placed in a 2 milliliter chamber with this and with the mesh in, so that it was protected from the rotating. Here they had short measurement periods and longer breaks because they had several chambers, but then they could calculate the slope between or within also. Actually, data were good enough for that. So a nice example of how you can handle such an organism is a very small volume of water. I'll also show an example where it's been used for microorganisms, ammonia oxidation kinetics in this microrespiration system. They were working with archaea and bacteria that oxidize ammonia. They had suspension of these microorganisms in the vials. They had a certain the injection lid, which is a lid with two capillaries, one for putting the sensor in and another where you could put a needle in and you can inject substrate as during the experiment. And that's what they used here. They had the ammonia oxidizing organisms in here. Oxygen was constant until they added 10 micromolar of ammonium chloride, and then oxygen consumption started immediately and then leveled off again when the ammonia was consumed. There was about 15 micromolar of oxygen that disappeared through this period. They were interested in knowing the stoichiometry of the ammonium oxidation. They had two options. It could either be two ammonia oxidize reaction with 3 oxygens, or it could be a 1-to-1 reaction between ammonia and oxygen. They measured here that there was 15 micromolar oxygen for 10 micromolar ammonium, so a 1 to 1.5 ratio, and that corresponds very well with this stoichiometry. So they argue that these microorganisms, they consume ammonia ions and not the ammonia gas or uncharged molecule. They also used this, so they used the oxygen consumption rate as an indicator of the ammonium oxidation rate after having established it here. And then they measured the oxygen consumption rate at different ammonia concentrations, and they could estimate Km values and Vmax values for these organisms as well. Yeah, so a very handy system for measuring both with micro and macro organisms. So the last part of this, I will move more towards the field with the aim tomorrow to be totally in the field with in situ instruments. This is also an application note, which you find on our website in under knowledge application notes, the winning formula. And this is written together with Ole Pedersen, who is professor at the University of Copenhagen, and he's been using microsensors for 25 years. Everywhere, more or less. He uses them in the field. He uses just a standard laboratory micromanipulator. He sprays it with silicone oil before he takes it into water, and then rinses it with fresh water when he gets back, and they live for many years for him. He's placed a pole in the sediment here so that he can mount the micromanipulator, and he can measure with a sensor around the roots of grass here. Then he also takes the same species into the lab, where he can go even more in detail with how oxygen is distributed in the roots. And he argues all the time that you should combine in situ and lab, because you can learn in the lab and explore and confirm in the field. So you can see what you have discovered in the lab and actually also makes sense in the field. And this is the PDF you could download from our website. I'll just show one data set from his work here. He's diving in a seagrass bed, placing oxygen microsensors. He has again this micro manipulator here, the sensors in the in situ amplifier for underwater use. And what he was measuring is he had a sensor in the water column, so measured oxygen concentration as a function of the time of the day. So from 3 in the afternoon one day to 3 in the afternoon the next day. And he was also measured inside the rhizome of this seagrass, and saw that in the evening, when it was dark, oxygen was consumed inside the rhizome and was totally anoxic during night. And then during when the sun came up again in the morning, oxygen would increase inside the rhizomes. And he uses the underwater meter for this. This is a diver operated meter, waterproof to 30 meters. Sensors are mounted via the in situ amplifier, and the meter here is operated using this stick that has a magnet on the end, so you can just touch the different parts of the instrument and you can operate it there, start locking and stop locking and so on. Okay, one more. Going to the field, this is Marco Fusi and his colleagues. They were working in the Red Sea in mangrove sediments, looking at the bioturbation by fiddler crabs. So they took this field multimeter and field motor out to the field. Splash-proof meter, waterproof motor, battery powered, and you can use this as a standalone instrument, so you don't need a PC. This is what I will show tomorrow in the demonstration, but I will use it with the PC. What they found was that they measured oxygen and redox into the sediment down to 50 millimeters depth. Black dots indicate bulk sediment, so far away from any burrow by the fiddler crabs, and the other dots here, the lighter ones, 5 millimeter, 10, 15, 30, and 45 millimeters away from these burrows. And if you look at this, bit difficult to see, but if you look at the light colored ones here, oxygen disappears more or less here, but then it shows up again here, it shows up again here and here. So, and the same if you go further away from the burrow, all the way up to at least 30 millimeters away. So what they argue is that oxygen is leaking out from the fiddler crab burrows and affecting the sediment. And they can really see that also on the redox. The black redox profile here is far away from a burrow, and see it goes very negative, whereas all the other ones, they are remaining positive actually all the way down to 50 millimeters depth. So these fiddler crab clearly affect the redox conditions around their burrows and in the sediment in general. And what they used was the field microprofiling system. This one can be used outdoors in the rain. The sensors are waterproof. The field motor is waterproof down to 10 meters, so are the sensors. Cables can be up to 10 meters long, and you can use them like this, operated from the keypad on the instrument, or you can operate it from a PC through our software. You can also use it as a two-dimensional system. So here an extra motor is mounted horizontally, so you can measure a series of profiles automatically. So you can make one profile, take out the sensors, move horizontally, and make another set of profiles and so on. Comes with this 6 sensor holder. We also had a 4 sensor holder, but you can also mount a micro manipulator on it if you want the opportunity to sort of easily manipulate the sensor horizontally here. So a very handy system for this. Yeah, just some details. I think I mentioned it most. Yeah, to say the battery has power for about 20 hours at 25 degrees centigrade. It has 8 channels actually, so you can have 5 amperometric sensors, 2 potentiometric microelectrodes, and 1 temperature sensor connected to it simultaneously. It can be used for this field stand, so you can take this spear off the base plate, take the motor off, and then you can drive this into the sediment or soil or whatever working, and mount the motor again, and you can use it there. And tomorrow I will get more into to getting how to get from lab to field and to deep sea measurements. That will be about at 2:30 tomorrow afternoon, Danish time. And with that, I will stop the presentation and hope you have some questions. Yes, please.
Yeah, so we have a raised hand by Olivia.
Yeah. Hello again. Thank you very much. It was very interesting. So I have this, the way you present it, it seems very, very, of course, it's great and seems very easy. So I have a question. You're doing experiments with developing microbial mats and in sediments. Of course, it's much better than just what we're doing right now, like measuring the headspace above with GC and trying to see the evolution of the gas. But how do you showed that it was kind of fragile. How do you protect the tip when you move around within the sediment so that you don't damage it? Because
well,
is that for that or?
Well, first of all you need to be careful, and then next, if you're working in the lab and you can see the sediment surface, then it's easier because then you can see if there's a stone or a shell or something lying around. You can avoid that. If you want to know if there's something hiding in the sediment, what I typically do is take a very thin needle mounted on a 1 millimeter syringe, and then sort of poke that into the sediment in the area around where I want to measure, not exactly where I want to measure, but just maybe 5 millimeters away around, just to see if there's a shell or a stone lying underneath the sediment that could be in create trouble when I start measuring. And if you do that, then very seldom you run into problems. It depends, of course, very much on your sediment. Yeah.
Would you recommend? I don't know. I've seen you have different way of protecting your tip.
Yeah.
Is there something to put around for this kind of application, or?
No, not really. Because if you want the high spatial resolution, so we generally say that you can measure and get independent data points at the same distance as the diameter of the sensor. So if you have a 100 micrometer sensor diameter, you can measure for every 100 micrometer. But if you use a needle sensor, which is 1.1 millimeter, then you can measure every 1.1 millimeter. And that using a needle sensor will also block access of, block the diffusion to and from the site where you're actually measuring the oxygen concentration. So therefore needle sensors are not really good. The glass sensors are clearly preferable.
Thank you very much.
Okay, then we have a question from Ketil.
Yeah, hi again. So when you showed the sensor holder that could hold 2 sensors, the one sensor seemed to be at an angle. Are you profiling with the sensor at an angle, and does that affect how fragile the sensor is or the stability of the sensor?
No, it works fine. We don't see problems with that. They are not very far from each other. You can actually adjust also the distance between the 2 sensors up at the sensor holder part. So the second sensor holder can be moved in many ways. So of course I'll always get the sensors as close together as possible at the upper end, so they are as parallel as possible at the bottom. But we don't see a problem in the one going in at a slight angle relative to the other.
It's not breaking more easily.
I don't think so. No.
Okay. All right. Thanks.
Yeah, you're welcome.
Yeah, the next one, Ulrike.
Yeah, so thank you. I don't have any experience yet with measuring hydrogen sulfide profiles. So if I want to do that and also measure oxygen profiles, you recommend to measure or to align oxygen, hydrogen sulfide, and pH sensors in this multi-sensor thing?
Yeah, well, you connect. There's only room for 2 sensors, so I would put the first the oxygen and the sulfide together, and then I would take the oxygen off and put a pH electrode on, and then yeah, maybe I would just keep the oxygen sensor on all the time, so I know where that's easier to get to find the sediment surface from the oxygen profile. So and then replace the sulfide with the pH electrode. So you have to measure them twice. You cannot get that in the same profile.
But in one of your, sorry.
Yeah, that's okay. Go ahead.
In one of your last slides, you showed this circular sensor holder to hold many sensors.
Yes.
I think we also have that.
Yeah, that one all sensors are parallel, so you cannot twist or tilt the sensors relative to one another. So there will be, so you can measure parallel profiles, but the tips will be relatively far apart. I think 2 centimeters or so. Yeah, so it would be
and that is not recommendable. Then that's too far.
Yeah, but it depends on how heterogeneous the sediment is. Because if it's very homogeneous sediment, it doesn't matter where you actually measure the profile. So then you could do it. Yeah.
Okay, and then the sensors are all the same length. So if you align them on the profiler, they would also be the same length then.
No, you will have to look at the tip, because they are handmade and pulled in the flame, so they will not be exactly the same length. There could be a centimeter or so variation, but you should place first put one sensor in, and then you put the other sensor in and place it vertically, so roughly oriented, and then tighten it, and then you can use the manipulation screws to twist it and move it back and forth to get it at the exact position. So that's relatively easy to do, but it's a good idea to have a microscope dissection microscope to look at the tips, or very good light. Then it's also possible by the eye.
Okay. Yeah. Thank you.
Hi. Hi. I was it's a very quick question. I was just wondering if there are stands you can do like horizontal measurements if you're having a biofilm on a rock wall, for example.
Yeah. Yes, that's certainly possible. Yeah.
Yeah.
Yeah, there's no problem. The only problem you can run into, if you point the tip upwards, then the bubble in the electrolyte may get up in the thinner part of the sensor and block electrical contact. So and then if you want to do that, we can modify the sensor when we make this. So we put a gel into the electrolyte so that the bubble will not get up there. So just let us know, then we can easily avoid that. But the standard sensor will just be a liquid electrolyte, because most people use them with a pointing downwards or not upwards. But horizontal is no problem.
Fine. So that was my only question. Thanks.
Yeah. Okay. You're welcome.
Okay, then we have Wright.
Thank you.
Yeah.
Yeah. My question is, I'm just trying to recall these calculations for gross photosynthesis and net photosynthesis. So if I get you right, you measure with light dark shift at a different depth in the sediment or in the mats, and that gives you the volumetric gross photosynthesis rates.
Yes, that's correct.
And then so how do you integrate that to get the area rates? And
then you have to you have a rate in
so
nanomoles.
Which depth do you start at here? I mean, in this profile, which depth do you start measuring? I mean, do you go in the mat just below the surface and start measuring?
No, in this case, I would want the 1, 2, 3, 4, 5, 6 intervals. Is that right? Yeah, between 0 and 1 millimeter. So the sensor has been in the center of each interval here, and then we assume that represents the interval thickness here. So that's why we can make a bar graph, so to say. So I would normally plot the bar graph so that the center is where so the depth center is where the tip of the sensor was. And then I would multiply the gross photosynthetic rate here, nanomoles per cubic centimeter per second, with the thickness of the layer, and then correct it up to per meter square and per hour. and then sum these up, these 6 bar graphs.
So you stop measuring till you get no.
Yeah, there has also been a measurement down here, yes, but there was no decrease in oxygen.
Okay.
No, that's right. Yeah,
okay.
yeah, and then it's just it's a matter of keeping the units right. Because you go from cubic centimeter to square meter, and you go from seconds to hours, and you go from nanomole to micromole. But that's
Okay. And another question is, I have done this a couple of times, and sometimes you end up having higher rates of net photosynthesis than gross photosynthesis when you do these measurements. Any reasons for that, or how do you correct for it?
Well, I don't know. I would expect that gross would always be larger than net.
Yes.
Yeah, I don't know. There may be a heterogeneity, and I don't know if the it depends on how you measure the oxygen profile, because if you can measure the oxygen profile, so you record the concentration of oxygen at the same time as you do the light dark shift technique, but that is not so accurate, because you will then need to get back to exactly the steady state. You'll get a more precise oxygen profile if you measure a profile by itself, and then you do this light dark shift by itself. And if that is not in the exact same spot, you may get a little bit of a difference. But I think that's up to experimental variation to give this different. That there should not be any systematic reason that you should get higher net than gross.
But do you think I should measure the oxygen profile with the same photosynthesis profile? I mean sensor. Sorry. Like I measure
same sensor is fine.
Sensor and then do the light dark shift right away.
Yeah, that's how I would do. So I'm sure that I have a very nice steady state profile, because if the profile is not quite in steady state, then the fluxes you calculate are not totally correct. Yeah. So
Thank you very much. Thanks.
You're welcome. Can I leave the underwater meter for 24 hours or even longer? Yeah, you can do that. I don't quite recall the battery hour. I think it's about 20 hours battery or so. It depends on the temperature, of course, but you can have an external battery as well, so just should be possible. Can the microrespiration system be used with other sensors like pH or hydrogen sulfide? Yes, they can be used with all our sensors. Similar to others. Is there a way to obtain dummy electrodes, cheaper electrodes, to test the mechanical resistance of the electrodes before running an electrode on a new sample type, for example, apple, soils, bark, etc.? Yeah, you can get that. Of course, we can make dummy sensors that will just be glass capillaries, or even old sensors we have that don't work anymore. So that's certainly possible. Is it required to have external reference electrode at the exact same depth of the potentiometric electrode? No, that's not necessary. You just need to make sure that there's a liquid contact between the tips of the reference electrode and the potentiometric electrode, for example, a pH electrode. So if you're working in a sediment core in an aquarium, you can put the reference electrode in one corner of the aquarium, and you can measure inside the sediment core, as long as there's a liquid contact. It becomes a problem if you're working in soil, for example. If it's not totally water saturated, then you may not get this liquid contact, and then the signal will start to drift. But in sediments, and it's no problem, just as long as the electrode, the reference electrode, is somewhere near the measuring electrode. Can I use the same microsensor in situ and in the lab, different connectors? No, well. A lab sensor cannot be used on the in situ connectors. That's not possible. But you can, and we have people doing that. They have an underwater meter, for example, they use as amplifier in the lab. So the sensors are in situ sensors. They are mounted in the in situ connector, in situ amplifier, and used in the lab. But you cannot, the lab sensors are glued together, and you cannot take that apart and move them between. Yeah. Thank you very much for listening. See you tomorrow.